the funny channel activity, that is the leakiness of the channels, increase, and it binds to beta 1 adrenergic receptors, which activate the cyclic AMP and increases the leaky channel open time. So it is causing more rapid pacemaker potential and faster rate of action potentials. So this is actually the myth, how the sympathetic activity is actually having the increased chronotropic effect, that is increase in the heart rate. There is increased in the dromotropic effect, that is there is increase in the conduction of action potentials. There is increase in the force of contraction of the heart, that is the inotropic effect. So whenever the sympathetic activity regarding the pacemaker cells that is being asked, you should have this thing in your minds, that the sympathetic nervous system is going to increase the chromotropic effect, the dromotropic effect, as well as the inotropic effect in the heart. Then we should be knowing about the function of parasympathetic activity. Now parasympathetic nervous system, the neurotransmitter is the acetylcholine. Over there we have learned the norepinephrine, epinephrine, and over here the acetylcholine. Now this acetylcholine is going to bind to the muscarinic receptors, the receptors of parasympathetic nervous system. So what is the function is that there is increase in the potassium permeability and there is decrease of the calcium permeability. So it is hyperpolarizing the membrane, the longer time to threshold, and there is the slower rate of action potentials. So we should keep this thing in mind, that there will be decreased chronotropic effect, that is decrease in the heart rate. There will be decrease in the dromotropic effect, that there is decrease in the conduction of action potentials. Then there is decrease in the inotropic effects. There is decrease in the contractility of the heart. Now the contractile cells, they have special aspects. What are they? There is presence of intercalated discs. Now intercalated discs, they have the structure, you can see by electron micrography, that there is presence of high convolutions and there is interdigitations. The joint adjacent cells with desmosomes and fascia adherens, they allow for syncytial activity. Syncytia means that they are working together, contracting together due to the gap junctions. So there is presence of more mitochondria in comparison to that of the skeletal muscle. There is less sarcoplasmic reticulum. The calcium also influxes from extracellular fluid, reducing the storage need. There is presence of larger T-tubules internally branching. Myocardial contractions are graded. Now the action potential of contractile cell. For that matter, the calcium ion is playing the major role. Action potential is longer in duration than a normal action potential due to calcium entry. Now the different phases of the action potential. The first one, that is the resting membrane potential in the contractile cells of the heart, they are minus 90 millivolts. Then 0, that is due to the gap junctions or conduction fiber action. Voltage gated sodium channels, they open close at 20 millivolts. There is temporary repolarization. Opening of calcium channels allows some of them to leave. Then there is plateau phase. Because of the voltage gated calcium channels, they are fully opened and they started during initial depolarization. Then there is presence of repolarization. The calcium channels, they close and potassium permeability increases as slower activated potassium channels open, causing a quick repolarization. You can see the difference between the skeletal muscle action potential graph and that of the cardiac muscle action potential graph. You can see over here, you can see the peak. Over here we have the plateau. The plateau phase prevents the summation due to elongated refractory period. So there is no summation capacity over here in case of cardiac muscle, and there is no concept of tetanus in case of cardiac muscle. So this table, in which we are going to learn about the comparison of action potentials in cardiac and skeletal muscle, is quite important. And what is it? It is about the membrane potential, which is around -70 millivolts in case of skeletal muscle. In contractile myocardium, it is -90 millivolts. In autorhythmic cells, it is -60 millivolts. Then we have the events leading to threshold potential. In skeletal muscle, the sodium entry via acetylcholine gated channels is responsible for this threshold potential. Over here, the depolarization enters via gap junction. In autorhythmic, the net sodium entry through the funny channels reinforced by the calcium entry. Then the rising of phase of action potential is caused by sodium entry in skeletal muscles, sodium entry in contractile myocardium, and calcium entry by autorhythmic cells. Then we have the repolarization phase, which is the rapid and is caused by potassium efflux. Over here, the plateau is by the calcium entry. The rapid phase is caused by potassium efflux. And autorhythmic myocardium, that is due to the calcium entry, the rising phase comes. Repolarization is caused by potassium efflux in skeletal muscles, by potassium efflux in contractile myocardium, and potassium efflux is the answer for autorhythmic cells. Then hyperpolarization, then duration is short in case of skeletal muscle, extended to like 200 milliseconds in case of contractile myocardium, and it is in autorhythmic cells, it is 150 milliseconds. The refractory period is generally brief over here, whereas in the contractile myocardium there is long refractory period. There is a resetting of sodium channels gates delayed until the end of action potential, and this is actually that there is no development of any sort of tetanus in case of the heart. So these are actually the basic comparative analysis you can say about the skeletal muscles, the heart contractile portion, and the 1% autorhythmic myocardium. Then we should be knowing about how this contraction process is taking place over there in case of contractile cells. Now, how the initiation? We know that the initiation is actually the function of that 1% of the heart cells, and which are the autorhythmic cells. So via the autorhythmic or pacemaker cells, the action potential is going to reach via conduction fibers, and the excitation contraction coupling, how it is taking place. It starts with the calcium induced and calcium release. Now action potential spreads along the sarcolemma. The T tubules, they contain voltage-gated L-type calcium channels which open upon depolarization. Now, when they open, the calcium enters into myocardial cell and opens the ryanodine receptors, calcium release channels. And this ryanodine receptors, they're going to do the release of calcium from sarcoplasmic reticulum, and there is actually the spark of calcium, the peak of calcium, and multiple sparks form a calcium signal. And then you can see that this calcium is going to do the contraction process you already have done. So how this is taking place, that via the action potential that is actually going to be spread over the sarcolemma, and then the via T-tubule voltage gated L-type calcium channels open. Upon depolarization, the calcium enters into myocardial cell and it opens a ryanodine receptors, calcium release channels, and the multiple sparks form a calcium signal. Now the calcium signal binds to troponin to initiate the myosin head attachment to actin, same as that you have done in the skeletal muscle, but the strength of contraction varies. The sarcomeres are not all or none as it is there in skeletal muscle. The response over here is graded. There is low level of systolic calcium, will not activate as many myosin-actin interactions, and the opposite is true. There is presence of length-tension relationships exist. The strongest contraction generated when the stretched between 80 and 100% of the maximum. Now, what causes the stretching? The stretching is caused by the filling of chambers with that of the blood. Now you know about the contraction, we should know about relaxation as well. Now calcium is transported back into the sarcoplasmic reticulum, and calcium is transported out of the cell by a facilitated sodium-calcium exchanger. Now as intracellular fluid calcium levels drops, interactions between the myosin and actin, they are stopped and the sarcomere lengthens. So this is how the relaxation is going to take place in this exchanger, which is sodium calcium exchanger, that is actually playing the major role. So what we have done up till now is the basic functioning, basic comparison between the contractile cells, the autorhythmic cells, and how the heart cells, they differ with that of the muscle cells, and how this excitation contraction coupling taking place, what is the role of calcium ion, what is the role of this threshold potential, why the plateau that is there, why there is long refractory period, and how it is important that there is no concept of tetanus over there in case of the heart. So we should be knowing about it. Now let's start with this coordinating activity taking place in the heart. What is that? That is the cardiac cycle. So the cardiac cycle is the sequence of events as the